Okay. All right. So welcome, Dr. Antonia Mey from University of Edinburgh. She's Chancellor's Fellow there in the School of Chemistry. Before that, she did a postdoc also in Edinburgh with Julien Michel. Pronounce that right?
Yeah.
And before that, she was in Berlin working with Frank Noe. That's where I know Antonia from, but I don't think we ever.
I don't think we overlap properly, right?
Yeah. Two times.
Yeah.
That's great. So, and you have an undergraduate and graduate degrees in physics and chemistry, is that right?
Yeah.
And yeah, so she'll be talking about proteins under the computational microscope from machine learning to molecular simulation. So we're very happy to have you here, and thank you for taking the time. And we're looking forward to see what you have to say today. Thank you.
Thank you very much for the introduction and also the very kind invitation. So I must admit, this is quite unfamiliar territory. I don't think I've ever given a presentation in the computer science department, so we'll see how this goes. All right, so I thought I talk about three things today. If we don't make it all the way through the three things, we'll definitely talk about the first two things. So the first thing is about how to use diffusion models for generating new molecules. And this is work a master's student's done in the group, and that was published last year. The next thing is about simulations and combining simulations and machine learning and how to predict binding affinities. These might be words that don't mean anything yet, but I'll explain them, so no problems there so far. And then hopefully we'll also talk a little bit about binding affinity predictions with machine learning. Yeah, so that's kind of the plan. But I thought we'll start with a bigger motivation. And how all of these three things actually connect together. So they all connect together through proteins. I did promise proteins under the computational microscope. So proteins are really the machines that manage life. They're basically involved in all molecular processes in our body, in plants, everywhere, really. And they take different shapes, they move in different ways, and are designed for specific tasks. So I'm just going to point out a couple here. So antibodies are there to help recognize disease. Enzymes such as trypsin help in your digestive tract. So they're literally everywhere. So one of the reasons why we as humans might be interested in studying them, other than just to understand how biology works at a molecular level, is also how can we manipulate them? How can we interact with proteins to stop a disease from occurring, or from maybe introducing a protein that doesn't exist anymore because of a genetic mutation and making it work again. So I thought I'll bring one example of where I think proteins are cool, and this is one of the topics I look at in the group, but I'm not actually talking about today, and that is an example from antimicrobial resistance. So this is actually, Jasmine Guven is looking at this problem. She is actually my first PhD student in the group, and what we're interested in is understanding how to avoid certain types of antimicrobial resistance. So antimicrobial resistance can occur in many different ways, but typically it's a way in which a bacterium is resistant to a drug. So if you think of this as a bacterium and you have penicillin binding protein, a beta-lactam antibiotic such as penicillin will bind this protein, and then the cell wall replication of the bacterium is inhibited, it dies. Great, right? So fantastic. The problem is bacteria have designed different resistance mechanisms, and one of them is through so-called beta-lactamases. And this is where naming conventions are great. So a lactamase seeks out lactam rings and actually hydrolyzes this chemical structure. And don't worry, we're not going to look at many chemical structures. This is as chemistry as it's going to get, probably. But basically, it catalyzes the breakdown of this ring, which means that the antibiotic is no longer effective. And as a result, well, we have antibiotic resistances. So one way of counteracting this is by designing new drugs that, for example, inhibit these beta-lactamases. And you can think of giving a combination therapeutic, which is a beta-lactamase inhibitor and your antibiotic. And one example of this is avibactam that was actually developed by AstraZeneca. So That's kind of cool. But in general, here is an example of how a small molecule can very specifically interact with a protein and regulate its function, and as a result, well, cure disease for us. And so I guess one of the big questions in drug discovery is, how do we come up with new ideas for molecules? If you think about molecules that are consisting of 50 atoms, typically probably have carbon, hydrogen, nitrogen, oxygen in them, the amount of possible molecules you can generate is enormous. So how do we find ones that are good? And then how can we test that they're good? And this is kind of a very naive way of looking at a drug discovery pipeline. Is you start out with some kind of molecule you want to generate, and we can discuss how we're generating that in a moment. Then you want to assess how well that particular molecule binds to a protein, and typically you want to optimize something like the binding affinity, but there are obviously various other properties you want to optimize as well. The one we're interested in the group the most is binding affinity. So this is a free energy of binding between a small molecule and a protein. And then you ideally want a computational method that is very predictive in the sense of that it can replicate experimental values well and can be used as a screening tool. In a sort of drug discovery terminology, you can say you have a hit or fragment hit identification. You have a starting molecule that you want to optimize, and then you kind of do an experimental validation, and this can take on whatever kinds of loops. I'm not claiming I'm doing drug discovery here. I leave this to the pharmaceutical companies, but I'm interested in methodologies that can allow us to make predictions at each of these stages effectively, and I'm going to tell you more about them. So how do you find a new molecule that binds a protein? So there's two main strategies in structure-based drug design, I think, which is high throughput screening and fragment-based design. So what is the difference between the two of them? In high throughput screening, you have a very large library of example molecules, and you use some kind of software tool to identify something that would bind a binding pocket of a protein. The problem here, it might bind loosely, but there are bits that could be optimized in terms of it doesn't fit the pocket so well, there's an unfavorable interaction or something like that. In contrast, fragment-based drug design basically takes a library of drug-like fragments, so they don't make up a whole drug because their individual affinities are too small. But you can basically get hits in terms of how a certain fragment might bind into a pocket. The problem here is, how do I go from fragment to drug? The problem here is, how do I optimize the molecule in the best way possible? What I'm going to now talk about is a generative model we've come up with that tries to address this question. So how to get from fragment to drug candidate, and this is work done by Nicholas Runchi, who's actually currently doing a master's project with AstraZeneca in Cambridge. All right, so what are the tasks you want to be able to accomplish in fragment-based drug design? So you have a bunch of fragment hits. Typically, you want to be able to grow a fragment into an existing pocket. You want to be able to link two fragment hits, or you want to be able to merge two fragments. And the way we wanted to address this is by using a diffusion model, and see how we could use a diffusion model to be able to do that. So I know I'm in a computer sciences department. You probably are much more familiar than my normal audience with diffusion models, but anyway, here it goes. So typically, well, it comes from image generation, I suppose, or learning, or yeah, generating images. So you start out with an image, and you want to be able to train a model that can generate new images. How do I teach a model to generate images in a diffusion process? So you start out with an image, and you iteratively add random noise to that image until you basically end up with white noise. You do this many, many times, and what you're trying to learn, or what your neural network is trying to learn, is that relationship between the noise and the image, and that's how you're training it. And this means now that if I have a trained model, I can give it any arbitrary noise, it will go through this denoising steps and generate me a new image. So that's great. Well, we're not looking at images of molecules, we're actually looking at molecules. So instead, we want to do the same process, but for molecules. And here's the point of it all. Basically, this what we've done is using a pre-trained equivariant diffusion model by Hogebaum et al. I'm probably butchering the name, I'm sorry. But the same idea applies, apart from when you think of molecules, you have additional constraints. You probably want them to be rotationally invariant. They're 3D objects, they're not 2D objects. So there's a bit more trickery you need to employ. But I suppose in some ways we were lazy and we just used an existing model. So that's great. So we have a model to generate molecules. How do we connect that up to fragments? And this is where iterative latent variable refinement comes in. So this is, again, something that is borrowed from images. So the idea is now, as you are doing your denoising step, if I introduce a reference, I can, in my already pre-trained diffusion model, I can push in my latent space my distribution towards a reference image. So you can see if I give Harrison Ford, I can generate images that look more like Harrison Ford and not just some other person. Okay, so we can now do this for fragment data using our molecular diffusion model. For this, we are not generating our own fragments, but during the COVID pandemic, the COVID Moonshot initiative, which was a, well, I guess almost global open source drug discovery initiative, managed to do a fragment screen against main protease. So this is the main protease, which is one of the drug targets for COVID. And you can see you have lots of different fragment hits. Now, if we take the fragments that attach to the main binding site, which is here, we're now trying to generate new molecules. So just again, sort of schematically, what happens is we have our diffusion process and we show a reference at each denoising step. And how much of that reference we show is dedicated by this thing we called silver rate, which is the selective iterative latent variable refinement. So we can basically tune how much of our reference molecule we're showing. So here's an example of what this kind of diffusion process looks like. So you've got 3 fragments, and basically we're generating this denoising step and making a video of it. And you can see how from our 3 fragments, we now have something that is merged together and fits into this binding site quite nicely. All right, so let's have a look at what we can generate and how that depends on the silver rate. So here we have our reference at silver rate 0, so we're not doing any conditioning at all. We basically get random molecules. They have nothing to do with the original fragments. And as you increase the rate, you can see that certain motifs are basically conserved in our generation. Okay, we can stare at a bunch of samples. That is not very informative. We want some data. So one thing we can look at is the root mean square derivation from our reference according to silver rate. And at 0, it's high. And the higher we push the silver rate, the more we resemble our reference. So that's great. The next thing we might be interested in, so far, we're generating molecules, but we've not given any information about the protein. Do we need to give information about the protein? So shape Gauss is a type of docking score. So a docking score kind of tells you how well a molecule fits into the binding pocket of a protein. And at silver rate 0, what you get is generate things that clash with a protein because we've not given any reference data. But actually at lower silver rates, we didn't have to provide any information of the pocket, but generally, because we're trying to merge structures that sit in the binding site, we can actually generate things that fit there. So we can do things like fragment merging, we can link fragments. So these are all nice operations. So far, we've generated a bunch of molecules, but are they any good? Do they actually have the potential of inhibiting that protein? Well, we probably want some kind of metric to be able to assess how good these molecules are. So first, we want to assess how good they are in terms of, can we make them in the lab? Are they synthesizable? How stable are they in terms of what is suggested? So these are measures you can get out of a Python tool called rdkit. So you can look at ratios of stable atoms and synthetic accessibility. So you want high ratio of stable atoms. So at some points it breaks down. So you want them to be more like up here. And synthetic accessibility is, I don't know, I don't love this score, but it's the best we have in terms of getting an insight. The lower, the better. So 8 is pretty bad. So whatever we're generating out of here isn't so great. And in fact, all of the things I've shown you just now, they look like real molecules, but most of the things generated are pretty crap. The good news is it's because it's the underlying diffusion model we chose to use. And we basically just used a model we could find in the literature, box down and pick it out of the hat and apply this method to it. We are now working on silver 2, which is basically a better version of this equivariant diffusion model. And we not only get much better synthetic accessibility, we can still do all of these other things, but we also generate molecules that can be found in a purchasable compound library. So we can actually buy molecules and could test this. Or, well, maybe not me, but people I work with.
You also need to change the silver rate, or is that?
Yeah, so basically the way, yeah, we basically have to do the same tests again. And I have some more data on this, but I thought for illustration purposes, I will keep it short here. So, right, so we can generate molecules, but we've not really got a handle on how good they are at actually interacting with the protein, other than shape-wise, they kind of fit into a binding site. So there's different ways of doing that. You can do something like docking. So there's I could give a whole set of lectures on docking. It's basically a tool that allows you to find a 3D conformation in which a protein and a ligand interact, and it might give you a score that is good or bad in terms of the interaction. There's many different algorithms, many different approaches to do this, but it's a general class of algorithms. Then you can do the same thing from dynamics, and this is the bit I'm going to focus on in a bit. Or you could try and do it through a machine learning model directly. I think now would be the time to ask questions on the first part, because I'm going to dive into the next part. Yes.
Yeah, so for the silver model, basically the issue that you are having, as far as I understand, is that you're training on a model that is trained on like QM9 or something like that.
Correct.
And then you're pushing it into sort of drug-like molecules. So you're way in the tail of the distribution.
Yeah.
You have probably more noise than you have.
Yeah.
So do you think that the way forward with silver 2 is to train on sort of like a more drug-like?
Yes, that's exactly what we've done. And also the other problem, so QM9 doesn't even have hydrogens. So actually, yeah, they infer them in some very dodgy ways. So yeah, there's basically a bunch of ways in which we've tried to improve the model by using a better data set and also different architecture, and yeah, lots of different things about it. But yeah.
I guess also avenue would be to sort of like make a guiding principle based on these other metrics like synthesizability and.
So actually what we're doing at the moment is rather than, so we know some chemistry, but somehow these models don't really incorporate chemistry. We know what a typical carbon-carbon bond length should be. So you can actually condition during the training, right? So this is a conditioning post-training, but yeah, we're investigating how we can condition within the training to make better unconditioned molecules and then use silver to condition those better unconditioned molecules.
Yeah.
And that seems to be so far working quite well. Yeah.
And I guess there's more question on the, if you can actually, like, what is the percentage of recreating all the fragments that are initially, like, because I guess technically it's not fragment linking, it's you're generating a molecule that mimics the fragments and is connected in a way. But I guess the root mean squared error is going down. But
Yeah.
Are you always able to recreate the original fragments? Like, are they always part of the molecule?
No, but you can basically the way, so at the moment I just showed data where you just have a uniform silver rate, but you could basically set a rate for each individual atom and you say, I don't want that atom type to change at all. And here I want things to change. And you can add additional dummy atoms that are not there in the original molecule, and they can then make up the linking space, although we had some issues with that. So in my head, this worked beautifully. In practice, this is work in progress, but yeah.
Can you say a bit more about that? Like, how does it work when you have the dummy atoms? Because you have to predefine what type of atom it's going to be before you do
No, you don't.
So do you predict in a two-stage process, like first the coordinates and then the identity of the
Yeah.
atoms?
Very much, yeah.
Is that the way it's done in the
No.
equatorial model?
No, it's not.
It's
Yeah. Yeah. Okay. All right. The next part is how do we know my model is my molecule is a 5-star molecule from dynamics? Got to get those 5-star reviews, right? Okay. For those of you who are unfamiliar with molecular dynamics simulations, I thought I will do a very quick slide on this. So typically what you do is you start out with some kind of structure. This could be a crystal structure. This could be an alpha fold structure, something. So it's basically, think of it as X, Y, Z coordinates of atoms of a protein. You then put these coordinates in a box and add lots more coordinates of your water molecules into that box. And then you apply what I would say is where the magic lies, but also all the problems, is a force field. So you want to evolve your protein according to Newton's laws of motion, and you somehow need to figure out what your force is or your acceleration. So you have your positions are from your atoms, and your potential you basically get from a sum of bonded interactions, angle interactions, torsions, and non-bonded interactions. And you can see there's a bunch of parameters you need to fit, and they will depend on your atom types and everything. So you have this massive sum, and you somehow need to fit that to a mixture of, say, experimental data or quantum mechanics data. And this allows you to define a force field, which can then be used to minimize your conformation. So that's like just a steepest gradient descent type problem. And then you can define the system you want to run. So do you want to run your simulation at constant volume and temperature or constant pressure and temperature? And there's different ways of regulating this. And at the end, you get something that looks a bit like this. It wiggles. So that's great. Like if you run it for a very long time, you get more wiggle. But how do we get something useful out of this wiggle that we can compare experimentally? All right, let's think about what we're actually trying to compute. So now, very artistic representation of a protein here in blue. And we're interested in wanting to understand how molecule binds and unbinds to the protein. You can define a rate, K on, K off, based on the binding and unbinding. Quite easy so far. This is related to the concentration of your ligand and your protein. And from that, you can actually define a free energy of binding of that ligand that is related to these K on and K off rates in a fairly straightforward manner. Great. So basically, all I need to do is simulate this a bunch of times. good to go. Right. Now this is where the problem with molecular simulations occurs, is that a typical drug off rate is in the 10 to the -4 per second. Our typical time step of our MD simulation is 2 femtoseconds because it relates to the fastest vibrations of our system. So maybe we can simulate 300 nanoseconds per day. Maybe we can push it to 500 nanoseconds per day. If we want to reach 1 hour simulation time, that would give us enough binding and unbinding events for most typical drugs. We're looking at, I don't know, 3 million years of compute. So we definitely need a better way of doing this. And thankfully, people over the last, I guess, 30 years almost, have been thinking about this problem and came up with something called alchemical free energy calculations. Okay, so I started working on these alchemical free energy calculations when I moved to Edinburgh, and my understanding of alchemy was this: we take lead and turn it into gold. Turns out it's not this, but I'm quite proud that I've now contributed my 21st century version of a alchemical process recipe with 2 best practices papers in terms of how to run these calculations. But I'm going to tell you how they work and what they are now. So, all right, going back to our problem, we want to understand how ligand binds to protein. This is super slow. But typically, in a drug discovery process, particularly when I talked about lead optimization, all my drug molecules look kind of similar. So you might have a core set at the center, and then you have only small changes elsewhere. So what we're really interested in is a difference in free energy differences between 2 molecules, so ligand 1 and ligand 2. So I can write this down. I still have the same problem. But what I'm interested in is this delta G ligand 2 minus delta G ligand 1. Delta G is a state function, which means it doesn't matter what route I go through in order to compute this free energy. So what I can do is artificially add linkers here where I say I'm going to change ligand 1 to ligand 2 while it's bound to the protein, and I'm going to change ligand 1 and ligand 2 while it's just in water. And that difference, bound minus unbound, should be the same as these differences and gives me the same free energy. Experimentally, I can't take that route, but in a computer, I can change the potential from that describes ligand 1 to the potential that describes ligand 2. And I can do this through so-called alchemical intermediates. This is where the alchemy comes in. And you have this lambda parameter that basically tunes whether you're in ligand 1 or ligand 2. All right, so now I can actually compute differences between 2 molecules. And typically, we're interested in many differences. So here's an example of a set of ligands. So this is a crystal structure. And now all of these black edges are these thermodynamic cycles I've just shown you. So there are a bunch of MD calculations, each of them. All right. Are they any good? So here's a typical benchmark. So we have a protein called tyrosine kinase 2. And this is what a typical ligand looks like for it. And you see there's this R bit, and the R bit takes 1, 2, 3, up to 16. These are the different R groups. And we have experimental values for them. So now we can validate our method and check whether we can actually capture our experimental values well. So here I'm plotting the experimental delta G versus the computed delta G using these alchemical free energy methods. And what you see is a very nice correlation. So I want ideally everything on this x equal y line. And we can look at some correlation statistics, such as the root mean square error, mean unsigned error, coefficient of determination, and things like that. So you can see where we're doing really well here. So that's great. So we've got, at least for this example, a pretty good method. We probably want to validate this on not just one protein ligand system. So here's a whole set of benchmark data. I guess we don't need to worry about exactly what they all are. But here I'm now showing you 1, 2, 3, 4, 5, 6, 7, 8 different proteins. Each of them has many different ligands, and how well we're doing in terms of mean unsigned error at predicting our binding affinity. So now each of these bars is basically one of these plots, but squashed into the mean unsigned error statistic. And well, the bit I was involved in is the red one. Obviously, I want to squint and say the red one is better than all of the other ones, but they're all pretty similar. So basically, there's a bunch of tools out there that are pretty good at predicting these binding affinities. So the tool we used are Flare and Open Biosim Space. And this is still work I did as a postdoc. So there's a bunch of tools that allow you to do all of these things. So you have commercial ones and academic ones, and I'm kind of involved in this one and this one and this one. So this is my spreading all my knowledge across all of the tools thing. All right, so at this point, we now have a method that pretty reliably estimates binding affinities. So we can actually tell whether a molecule is going to be 5-star or not. The problem is, I said, this method is better than the one that is impossible because it would take 3 million years, but it still takes 3 to 4 hours to get an estimate. And if you want to do that for many, many, many ligands, that is expensive. So what we actually want to be able to do is do something like active learning that allows us to find ways of labeling our data effectively. So we have typically a set of compounds that we want to test against a protein, maybe multiple proteins. So we can call that a ligand pool. And now we want to define an active learning cycle that allows us to pick effectively from this ligand pool without having to actually run alchemical free energy calculations for each of these ligands in there. So we pick a subset, we label them. So the labeling could be these alchemical free energy methods. It could be a different method, but for alchemistry, it makes sense because, well, it's expensive, computationally expensive. And then we basically train a surrogate machine learning model that then helps us select the next batch of molecules to pick a new subset and so forth. So we're doing this active learning cycle. And so this is work that was done mostly by Juan Garrahn, who's a PhD student in my group. And he did this as part of his internship with Excensia. So these are my collaborators at Excensia. Yeah, so I'm going to tell you a little bit about the findings we have. So the idea was, how effective is an active learning method in terms of labeling and picking out our ligands, and what is the best set of parameters we want to choose for our active learning? So the things we tested were, for this machine learning model, we used a Gaussian process regression and a pre-trained internal chemprop, so it's a machine learning model for property prediction, basically. In terms of strategies, so you can, in your active learning, you can randomly pick, or you can use an exploration or an exploitation phase. So for exploration, we basically pick new samples that have high uncertainty based on our chemprop or Gaussian process model. Or for the exploitation, we basically try and label points that have predicted high potency based on our model. What we were trying to do is say, okay, we have, I don't know, 6,000 compounds, and we want to select 360 for labeling. So we are willing to run 360 alchemical free energy calculations, but it's a fixed cost. So what is the best strategy to use using these compounds? So what are the data sets? We use publicly available data sets. This TIC2 data set is a great benchmark. It's, yeah, everyone's been using it. And then you have, yeah, a bunch of other data sets. The point is, we basically tried a variety of data sets in terms of how many ligands there were, what's the variety of measurements, and how many top binders there would be in 360 compounds. So these are the affinity distributions, and then you can see, if you squint and do a UMAP projection, you can see clusters of compounds that basically relate to these scaffolds or these similar ligands I talked about. I must admit, we didn't actually run any alchemical free energy calculations, so we just used the experimental data available because we had that data, so it was easy to do that. And here's an example of the top 2% of compounds in terms of affinity are distributed mostly up here and less down here. So the types of things we were looking at was recall, Spearman rank, coefficient of determination, and root mean square error. So there's a lot here. I think I'm gonna probably focus in on our 2 and top 2% recall. So we have our data sets. We want high score for rank, and we have our Gaussian process. Ignore the Gaussian process. Chiral is basically the same thing, and our ChemProp model. And what you can see is mostly they kind of perform similarly, apart from when our data sets become small, and then the Gaussian process actually performs better than our ChemProp. And in terms of top recall, the more compounds you have, the better it is. And for Emperor, for some reason, there is some variability in terms of how they perform here. Then the next thing in terms of protocol, do you just go random exploit, random explore, exploit, random exploit? So most of them are all doing better than baseline, so that's great. So if you just randomly select, not so great. Random explore and exploit works well for large data sets. Random explore, exploit is better for small data sets, I guess, at least from the limited data we have. We also wanted to look at noise. So we basically added noise on our labeling and see how that would change our ranking. So moderate noise wouldn't change it massively. If you add lots of noise, the model wouldn't learn so well anymore. So that isn't that surprising. And I think the key take homes, so we ran a lot of benchmarks. If you're interested, there's a lovely paper on it. But I think the main takeaways we found is that Gaussian process models are actually pretty good. They're cheap to train and easy to use. And I think the main thing, active learning can actually really help navigate large ligand libraries and select out compounds for labeling, either through experiments or through an RBFE calculation. So that's the end of this part. Yes.
Go to slide 27. I was just curious about this MPRO data set. So do you think that this that the big difference in the performance is related to the different, like that maybe the scaffolds are more, or the compounds are more similar to each other in the other data sets, and it's easier to learn, and an MPRO is just like completely different compounds? Is that?
Well, they're not completely different. So they also ran alchemical free energy calculations for them, but they're definitely more diverse than the TIC2 data set. It's basically a really clear core scaffold and then lots of variations of that. And the Emperor is also the smallest data set, so I'm a bit like, I'm not super trusting of any of the results from Emperor, but we ran it anyway. Yes.
The way I understand is that you want to kind of extend this in a way, how would you expect? You have a, let's say, budget of free energy calculations you can run, right?
Yeah.
And you want to maximize the CP information.
Yes.
But so I'm not the FEP person, so
Yes.
I don't know a lot about it, but I feel like isn't the one big problem in FEP that different docking, they can just define a docking grid, that it's very difficult, like it's very difficult to set up like a workflow that always works for each ligand to kind of start the FEP one, or is that?
No, so that used to be the case, but I think in the last sort of 5 years, at least commercial tools work like this. There will be some things that fail, but I think at this point, if you get to this point, you would have done a trial run with similar compounds for this particular protein. So, you know, you tried, I don't know, 20 compounds, they seem to work fine. Then it should be able, like, there should be very little reason why large chunks would fail. I mean, you will never have 100% perfect performance, but you're happy, like you'll accept that there'll be some failures. Yeah. Okay. All right. I think I have time to talk about the last thing. So last thing is about predicting these delta Gs directly from data using a machine learning model rather than using simulation. And this was motivated basically when I started my group. I was like, oh, loads of people are reporting these machine learning binding affinity papers. Why do I bother running these really complicated, expensive simulations? Can I just abandon them and maybe start working on just machine learning models instead and focus on certain applications or, yeah. So basically I got curious and tried to understand what is the landscape of these models, what are the architectures, what are people doing, and how do they learn to kind of get a better understanding. And this is, again, work done by Rohan, who is in my group. And effectively, what we've come up with was a sort of consensus architecture that is similar to some of the best performing architectures in 2021, 2022. So there might be better ones out there now. And these were graph convolutional atoms that will and then connect them up in an adjacency matrix. That is called a contact map in the protein world. And then from that contact map, you can define a graph. You can use a graph neural network to then basically get a protein encoding out. You can do the same thing with ligands. So even if you have a 1D SMILE string, so this is a 1D representation of this 2D structure, you have connectivity. So your atoms are connected by bonds, and that can be reflected again in your graph. But then in addition, rather than just having the connectivity, you can also give it node features. So node features would be things like aromaticity, degree of an atom, number of hydrogen bonds, and so forth. So again, you can encode your ligand in this way. So now the idea was, what if we iteratively change some of these things? How does that change the output in our binding affinity using this particular machine learning architecture? So for the ligand encodings, we just use the original ligand graph. We do something called a random point randomization, which I guess doesn't really matter that much. The one that is interesting is basically the random node features. So basically, we keep the graph, but just change our node features, and then just randomly sampling our ligand graph. So basically, we have iterations of perturbations from the original graph to basically completely random graph. And for the protein, we use 4 different ways in which we encode our contact map. So we use an AlphaFold model. I mean, we could use a crystal structure, but we didn't have enough crystal structure data. So we use an AlphaFold model and computer contact map. We use PCONS4, which is a model in literature, and ESM1B, which is this transformer-based language model. And then just a random encoding as a sort of test. What is the data sets we use? Well, kinases are a class of proteins that are relevant to, well, all sorts of illnesses, but probably most famous for cancer research. And what is interesting is there's a whole kinome out there, and they're all kind of similar. So one of the issues with kinases is selectivity. So you want to be able to selectively target one kinase, but not another. So having good, reliable predictions for binding affinities for kinases is useful. And the other thing is, because it is a cancer target, there's a lot of data available. So basically, there's 2 data sets, data set 1 and data set 2, which have varying amounts of binders and non-binders. So typically, the number of binders is much lower than non-binders. But yeah, these are the 2 data sets we used. And then in terms of what did we do? So the first thing was look at how good are our contact map prediction methods. So we have true contacts in turquoise, lost contacts in orange, and in pink, we have false contacts. So AlphaFold, so this is an example where we compare against a true crystal structure, is doing really well. Pecons 4 is also doing pretty well, ESM less so, and thankfully random is doing terribly because that's what I would want it to do. If we now do this for all of our proteins we have crystal structure for, we can actually look at some statistics. So here we mostly, I think the interesting one is Matthew's correlation coefficient. So 1 is what we're interested in. So ESM has got very little correlation, and sort of AlphaFold and Pecons 4 do similarly, but they have this weird tail. And we tried to investigate what was going on, but it's a bit unclear. Yeah. All right. So basically we have 2 pretty good contact map predictions methods and 2 not so good ones. Matthew's correlation coefficient. Okay, so now if we take these different Pecons 4, ESM, and AlphaFold and put them in our neural network architecture and try and do the affinity prediction, we get a really high correlation, Spearman, low root mean square error. So what we want. If we do the same thing for random, we have that same problem. I mean, not the same problem, we have the same results, so that's not very good. For data set 2, it's a little bit better, as in there's a small drop for the random, but still random is performing rather well. So that tells us that whatever protein encoding we're using is probably not getting much information at all from it. So yeah, if people claim that these types of architectures are good at predicting things, I'd be hesitant. Yes.
No, I was just thinking, like, is this model trained end to end? So you train, okay, yeah. So basically the problem here is that the representation of the model is informative.
Yeah. If we now look at changing our ligand encoding, we do get a significant drop for the random node and random ones. So basically, we might as well just get rid of the protein part of our architecture and just train on the ligand, which is obviously not what we want. So yeah, this was an interesting finding and kind of made me happy because it meant like these models aren't so great yet. And also means I'm always slightly suspicious of new models that are just slight derivatives of these current ones. Having said that, we've now been working on a new and improved version where we are showing that we can do a combined encoding that actually seems to work quite well. But then also our overall performance drops. So yeah. All right, and with that, I'm concluding. So the main take-homes hopefully from today is that diffusion models can be used to generate new molecules that fit into a protein binding site by using silver and the selective iterative latent variable refinement. Alchemical methods, these simulation-based methods, are really good at having a decent predictive accuracy for binding affinity predictions, but are still quite computationally costly. So using active learning in combination with them is a nice way of going about. And then, yeah, better approaches are needed for protein ligand binding predictions, at least in terms of the architectures we looked at. And yeah, thank you very much for listening. Just want to point out Rohan, I briefly mentioned Jasmine and Nicholas who did the work. And yeah, thanks for listening. Mm-hmm.
So towards the end of your talk.
Yeah.
Sorry, moderating after. So towards the end of your talk, you had the example with I also asked a bit, but this about the protein representation. So how much do you think that this is a problem that you are taking like a potentially huge representation, right? And then you're collapsing it? I guess you do some sort of in your graphing level, you're doing some sort of aggregation at the end, and you have some sort of like dimension a vector which is like some maybe 100-dimensional vector that is aggregating their whole protein graph into one vector, right?
Yeah.
And maybe in the protein ligand complex, there are maybe, I don't know, 5% of the atoms that are that representation that actually contribute to the interaction. So do you think that there's some maybe smart way of maybe like selecting out the atoms that are close to the ligand?
We did this actually. I didn't show the data here. It's part of the paper. So there's a data set called CLIFFs, which basically selects out all the amino acids that interact with the ligands. And we just trained CNN on that. And they perform comparatively well to the 2D models in terms, like, as in, like, the performance was similar, but they still had exactly the same problems in terms of, like, while we gave the specific residue interactions, I think the issue is how we concatenate ligand and protein vectors. I think the way people have been doing it is dumb, because you just go vector, and then fully connected network for your prediction. And I don't think that makes a lot of sense, because you don't give the correct weights for the important parts, really, because everything's equally important.
Right. So basically you throw away the information about the specific interactions that you have.
Yeah.
For that same model, did you also benchmark against other kind of protein ligand fingerprints, like the CIF?
Yeah.
Did you compare to those, or I guess I'm curious how it would perform in that case without using a deep learning approach. I don't know how good those fingerprints are.
We didn't look at this. No, we did not. I'm trying to, so we have subsequently been looking at fingerprinting a little bit, but now we're actually using a Cambridge model, which seems to be doing better than just ECF fingerprint or Morgan fingerprint or something like that. Yeah.
And sorry, just for the the connection.
Yeah. Concatenation, yeah.
No, sorry.
No.
Just for the protein representation.
Ah, yes.
The matrix. I forgot
the contact map. Yeah.
This is basically ligand information free, right? It's just and is it amino acid, like in the sense of just the connection, or does it also contain the type of amino acid?
No, so you also have node features, so it would contain additional node information on hydro. I think there's like 80 parameters or something like that. Like, is it a hydrophobic amino acid? Is it a blah 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 thing? Yeah.
And I think the problem, I guess, with kinases especially, is like you have like the ATP binding site, which like all the like most known inhibitors bind to, but then you also have like it's 30%, which is just extracellular binding or something. I think that's why I guess you would hope that by including the whole protein you can predict both, but I think having maybe just as maybe suggest like having the smaller just the pocket, I think might be just having like I don't know how you would do it, but
yeah, but I think so. I mean, so as we were working on this, someone else published a similar paper looking at methods for 3D complex representations, and they did a similar thing where they found that if we didn't include the protein in our complex data, it basically doesn't really change the performance of the model. So yeah, I don't. So I think, and then there was a very recent paper by Andrea Falkeman's group where they did enrichment of protein complexes with docking, because one of the problems is why we're not using more 3D posers is because the data isn't there. So we need to somehow enrich the data with a more or less reliable other computational tool. So.
I'm not sure, but I don't also have a paper recently out also about protein, kinase ligand pocket interaction fingerprint as well. I don't know if
Yeah.
it's something similar.
Yeah. I think they were using that for their complex stuff, but I've not looked into using it. I mean, basically we've only been looking at 1D or 2D methods. So either based on, well, CNNs, and now we're using language models. And that seems to work a little bit better. But actually the other thing people are really bad at is, so you get in these models, like your correlation, like all of these metrics are really high because your data set is not split in a sensible way, right? So you don't capture generalization of these models particularly well because you have bleeding of different target classes, different scaffolds into your test and validation sets. So if you do sensible splits, such as a drug, a target split or a scaffold split, you're gonna get a drop drastically, not necessarily with these methods, but with some other methods. Actually, no, also with these methods, but yeah. Sorry, that was very drug heavy, but.
So I'm curious about the active learning approach.
Yeah.
So how big of a performance do you think that we can push these things? So I see that you fixed the budget, right? And then you compared them against each other with
Yeah.
Like, what do you think, like, if you had a floating budget, like, how do you, I guess it would be very case dependent, but like, what would you say sort of an expectation, the number of actual alchemical calculation you need to do to get a sort of a reasonable handle in a sort of a real case scenario now that you have a bit of experience with?
I mean, I think about, doing about 400 calculations will probably be good enough in a lead optimization for training a Gaussian process model. You'll definitely do better than random. and then I think, well, you could try and optimize this to death, but I think in a real discovery scenario, it's good enough. You're picking enough top 2% binders, but then this is a retrospective study, so
you don't know what you don't know.
Exactly. So yeah, and I don't think, well, Extensia was not very happy to just let us burn this on, like, test this and burn some large amounts of compute time on a active drug discovery case. So I think they are now trying to use this, but more sparingly, which seems a bit dumb because
I guess they also have some sort of initial condition dependency, right? When you're starting with a single lead or maybe one or two leads, and then you just train a model on this, and then, but do they converge, do you think, generally into similar things?
I don't know. No.
Because I mean, there's a big sensitivity to the initial condition in these active learning, because I mean, it's not a process that we expect to be like agoric in general.
No, but I guess if you have a couple of hit molecules and you don't have too many scaffold hops and do an enumeration of R groups that are sensible and generate a library like that, you can probably quite effectively pick things out using this approach. Anything else? Don't know. We haven't looked into it, but my guess would be less reliable. No. I think it's a sensible question, but I think, we've not really looked at I think the problem is the data sets, we were very limited to data sets that were publicly available, and apart from, like, they all have different issues with them effectively. So yeah.
Okay, so let's thank Antonia again. There's for those